Hey everyone, my name is Aissvarya Shankar from University Putra, Malaysia. Today I will be presenting on my research entitled Identification of Differentially Expressed Genes in Dupuytren's Contracture, a study based on geodatasets. Dupuytren's contracture or DC is a fibroproliferative disease of the palmar fascia characterized by flexion contractures in the digits and associated to an aberrant buildup of type 3 collagen. DC is a complex disease with both hereditary and environmental influences. It is suggested that the disease is inherited in an autosomal dominant manner. Some of the factors that can contribute to the disease progression include smoking, alcoholism, manual labor, and other underlying conditions such as diabetes and epilepsy. However, DC currently has no known cure and the specific etiology is still unclear. Whereas differentially expressed genes or DEG is a way to understand etiology of diseases and eventually to treat them. Knowledge of DEGs can aid in the discovery of causative genes and gene pathways for a disease of interest, and in our case, DC. Thus, in this study, we aim to understand the genetic etiology and the molecular mechanisms leading to the development of DC through DEG analysis. For our methodology, we use Affymetrix microarray datasets from the GeoDatabase. These datasets were pre-processed and normalized using the LimaR package. MDS plot and box plots were constructed to visualize if the data were normally distributed. The studies that were included in this analysis were based on these criteria, which are Homo sapiens within 20 years range, have at least 2 groups, studies involving genes and DC, microarray studies, tissue samples, and must have a control group. Next, the different lists of DEGs were integrated into a robust list using the robust rank aggregation RRA algorithm and cutoff values of P lesser than 0.05 and log2 fold chain values of more than 2 or lesser than -2. This would be our first list of DEG. Then, a network analysis was constructed from this list using the string and cytoscape software. The plugins of the software CytoHubba and CytoNCA were used to develop list of DEGs that were subjected to the Venn diagram analysis. This would be our second list of DEG. Lastly, M-code was used to determine gene clusters from the network, and this would be our third list of DEGs or our hub genes. All 3 lists of DEGs, which are integrated DEGs from RRA, common DEGs from Venn diagram analysis, and hub genes from M-code, were subjected to gene anthology and cake analysis. As a result, the PPI network of the 165 integrated genes showed 144 nodes and 79 edges with different interactions between the DEGs. Some of them were connected while some of them were not. The Venn diagram analysis, on the other hand, from the CytoHubba and cytoanalysis, gave us a total of 33 overlap genes. These genes were further analyzed using gene ontology and cake. Furthermore, from the PPI network, 2 clusters consisting 6 genes in total were derived. These genes are HADH, FASNs, GCK, CTNNB1, CTSK, and CDKN2A. The possible involvement of biological processes and pathways were further elucidated through enrichment analysis. The enrichment analysis of the integrated list of DEGs, which is our first list, showed that the most enriched biological processes are canonical Wnt signaling pathway and cardiac muscle cell development, while the most enriched pathways are Cushing syndrome and hyposignaling pathway. Similarly, the enrichment analysis of the common genes from the Venn diagram analysis demonstrated that the most enriched biological processes are canonical Wnt signaling pathway and regulation of canonical Wnt signaling pathway, while the most enriched pathways are hyposignaling pathway and Cushing syndrome. It has been reported that Wnt signaling pathway may be involved in the development of DC as well as other fibrotic diseases through an accumulation of beta-catenin, causing uncontrolled proliferation of myofibroblasts and inductions of genes involved in collagen remodeling. Targeting the Wnt signaling pathway and downstream proteins could potentially lead to the development of treatment of the disease. For the hub genes, the King analysis showed that the most enriched pathways are insulin signaling pathway and Cushing syndrome. Previous studies have reported that diabetes is a risk factor of DC, and individuals with diabetes have a higher possibility of developing DC. Overall, the hub genes is GCK, HADH, FASN, CTNNB1, CTSK, and CDKN2A identified in this study possess high biological importance in diseases such as diabetes, epilepsy, and fibrosis, which are all risk factors of DC. Our in silico analysis correlated with previous findings where Wnt signaling pathway may significantly contribute to the development of DC. And the analysis also revealed other pathways and syndromes such as hyposignaling pathway and Cushing syndrome that have been less explored in the context of DC. DC. Further exploration of these genes could facilitate in the development of potential biomarkers and therapeutic targets of DC. Below here are the references used for this poster. I would also like to acknowledge the Ministry of Higher Education, Malaysia, in financially supporting this study. That is all from me. Thank you all for listening to my presentation.